Hello everyone, and welcome to the first and only mini lecture for Module 3 this week. And in this mini lecture, we will talk about eukaryotic cells. Robert Hooke, a microscope enthusiast and a very colorful character, coined the term cell for the box-like structures he observed when viewing cork tissue through a lens. 2 centuries and many advancements in microscope construction and lens advancement later, a botanist named Matthias Schleiden and a zoologist named Theodor Schwann proposed the unified cell theory, which states that living things are made up of one or more cells. The cell is the basic unit of life, and new cells arise from existing cells. You've read about prokaryotic and eukaryotic cells. Prokaryotic cells are pretty simple to understand, so I will take this opportunity to go over eukaryotic cells in a little bit more detail. Here we see an example of an animal cell here on the left and a plant cell here on the right. In the next few slides, I'll take a moment to point out some key organelles. Let's look at eukaryotic cells now, starting from the outside and working our way inside. We'll start with the plasma membrane. Both prokaryotic and eukaryotic cells have a plasma membrane. The plasma membrane is a phospholipid bilayer with embedded proteins that separates the internal contents of the cell, which would be on this side, from the surrounding environment. And we can see the blue, green, yellow protein structures here. A phospholipid is a lipid molecule, and you remember those have 2 fatty acid chains and a phosphate containing group, which is the R group. So the 2 fatty acid chains are these yellow strings hanging here, and the phosphate group is this orange bubble. See, so already we talked about proteins, we've talked about lipids, and we've discussed 2 macromolecules that we discussed in Module 2. The plasma membrane controls the passage of organic molecules, ions, water, and oxygen both into and out of the cell. Waste such as carbon dioxide and ammonia also leave the cell by passing through the plasma membrane. In Module 4, after we get back from spring break, we'll take a detailed look at the plasma membrane and its role in regulating what happens in the cell. The cytoplasm is the cell's entire region between the plasma membrane and the nuclear envelope. We can see it here with this kind of yellow tinted area and this green area here. The cytoplasm is comprised of organelles suspended in a gel-like cytosol, the cytoskeleton, and various chemicals. Even though the cytoplasm consists of 70 to 80% water, it has a semi-solid jello-like consistency because of the proteins that it contains. Many metabolic reactions, including protein synthesis, take place in the cytoplasm. Now we'll look at the nucleus. Typically, the nucleus is the most prominent organelle in a cell. The nucleus houses the cell's DNA and directs the synthesis of ribosomes and proteins. So let's look at it in more detail. The nuclear envelope, which is this first, this dark purple thin layer here, that's the nuclear envelope on that. Oh, so this is a nucleus, this whole circle, and the dark purple is the nuclear envelope. The light purple is the inner portion of the nucleus. So the nuclear envelope is a double membrane structure that constitutes the nucleus's outermost portion. Both the nuclear envelope's inner and outer membranes are phospholipid bilayers. The nuclear envelope is punctuated with pores, and we can see those here. These little spaces in between these blue things, those are pores. So the pores control the passage of ions, molecules, and RNA between the nucleoplasm, which is this area here, and the cytoplasm, which is out here. The nucleoplasm is a semi-solid fluid inside the nucleus, kind of in the same mind as a cytoplasm, and it's where we find the chromatin, which are these loopy structures here, and the nucleolus, this dark purple circle. You may remember that in prokaryotes, DNA is organized into a single circular chromosome, but in eukaryotes, chromosomes are linear structures. Each eukaryotic species has a specific number of chromosomes in the nucleus of each cell. In humans, the chromosome number is 46. Chromosomes are only visible and distinguishable from one another when the cell is getting ready to divide. When the cell is in the growth and maintenance phase of its life cycle, which is most of its life cycle, proteins attach to the chromosomes. You can see them here. These blue circles are the proteins, and they resemble an unwound jumbled bunch of threads. We call these unwound protein chromosome complexes chromatin. You can see that here. Chromatin describes the material that makes up the chromosomes, both when condensed, like we see it here, and decondensed, like this structure here. Now let's look at ribosomes. Ribosomes are the cellular structures responsible for protein synthesis. When we view them through an electron microscope, ribosomes appear either as clusters called polyribosomes or single tiny dots that float freely in the cytoplasm. They may be attached to the plasma membrane cytoplasmic side or, as seen here, attached to the endoplasmic reticulum cytoplasmic side and the nuclear envelope's outer membrane. Ribosomes are actually large protein and RNA complexes consisting of two subunits, the large one seen here in orange and the small one as seen here in white. Ribosomes receive their orders for protein synthesis from the nucleus where the DNA transcribes into messenger RNA. You can see the messenger RNA strand moving into the ribosome here. Because protein synthesis is an essential function of all cells, including enzymes, hormones, antibodies, pigments, and structural components, there are ribosomes in practically every cell. Ribosomes are particularly abundant in cells that synthesize large amounts of proteins. For example, the pancreas is responsible for creating several digestive enzymes, and the cells that produce these enzymes contain many ribosomes. This is another example of form following function. And here we have mitochondria. Mitochondria are popularly known as the powerhouse of the cell. They are responsible for making adenosine triphosphate, or ATP, the cell's main energy-carrying molecule. Mitochondria are oval-shaped, double-membrane organelles that have their own ribosomes and DNA, and each membrane is a phospholipid bilayer embedded with proteins. And here we have some catch-all. So peroxisomes are small round organelles enclosed by single membranes. They carry out oxidation reactions that break down fatty acids and amino acids. They also detoxify many poisons that may enter the body. So an example of structure and function is that the peroxisomes in the liver, their main role is to break down alcohol to prevent alcohol poisoning. And then we have vesicles and vacuoles, which are membrane-bound sacs that function in storage and transport. Vesicles and vacuoles are virtually indistinguishable from each other, but vacuoles are larger than vesicles, and vesicles can fuse with the plasma membrane or other intracellular membranes. At this point, you know that each eukaryotic cell has a plasma membrane, cytoplasm, a nucleus, ribosomes, mitochondria, peroxisomes, and in some, vacuoles, and we haven't even talked about the endoplasmic reticulum and the Golgi. But there are some striking differences between animal and plant cells. Centrosomes are unique to animal cells. You can see them here on the left. They are microtubule organizing centers that play a big role in arranging chromosomes during cellular reproduction. Animal cells also have lysosomes, which are sort of in the same category as peroxisomes, vesicles, and vacuoles. Lysosomes are the cell's garbage disposal. They contain enzymes that help break down macromolecules. Lysosomes have a low pH that, if you remember from Module 2, helps speed up reactions like the breakdown of lipids. In plant cells, all disposal takes place in vacuoles, so plant cells don't have lysosomes. Plant cells instead have a cell wall. The cell wall is a protective structure on the outside of the plasma membrane that provides structural support and gives shape to the cell. And that's what this purple arrow is pointing to, this dark green stripe here. That's the cell wall. The plasma membrane would be the light green area. Some prokaryotic cells have cell walls as well, as you may remember from the reading. Plant cells also have chloroplasts, seen here in this purple box, these green structures. And if you remember from General Biology 1, chloroplasts are the site of photosynthesis. So that brings us to the end of this little lecture. Make sure that you have filled in all the spaces in your graphic organizer. You'll need this information to complete the rest of the module.